Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. This is our third and final biochemistry lecture in the endocrine module. We will continue our talk on mechanisms of hormone actions. We will focus in this lecture on cell surface receptors. Although we talked about some of these receptors before and their mechanisms of action and how they transduce signals, that is how a signal is transmitted from one molecule to another, this lecture is sort of nice because it wraps up all of the information that we have discussed before into a topic, into something that is practical. So let's go on. We'll start with G-protein coupled receptors. You know how they function. So G-protein coupled receptors are 7 transmembrane domain receptors. So they're composed of 7 transmembrane domains, and you have portions of the protein extended to the outside of the cell or to the inside of the cell. All right? So there are different types of receptors, and actually these receptors are really important because let's say that I think it's about 40% of drugs target receptors, and that's a large percentage. 25% of drugs overall target G-protein coupled receptors. So these proteins are really important. They have high degree, although they function similarly, there is still difference, or there are still differences in terms of homology among these receptors. So if we consider the beta-1 and beta-2 adrenergic receptors, they are only like 50% identical rather than being, a high homology. The alpha and beta adrenergic receptors have even less homology. But overall, there are 2 important domains or segments of these receptors that are important. One of them is the ligand binding domain, which is extended to the outside, and we have also to the inside the G-protein binding domain as well. So they have homology in terms of that, but differences as well to specify the type of G-protein that binds to the receptor. All right. So there are different types of G-proteins inside. Remember that these are heterotrimers. They are composed of 3 subunits, alpha, beta, and gamma. Okay, that's alpha, that's beta, that's gamma. The alpha subunit is the important functional subunit. The beta and gamma subunits are regulatory subunits. So there are different types of G-proteins. We have G-alpha proteins, and they can be stimulatory, or they can be inhibitory. So you have the G-alpha S for stimulation or G-alpha I for inhibition. We also have the G-alpha Q. The differences between them here is that the G-alpha stimulatory, it acts on beta-adrenergic receptors, different types of ligands, and you have stimulation of the adenylate cyclase, hence the abbreviation of S. Now, the G-alpha I inhibits the adenylate cyclase, as we will see in a few minutes. Now, the G-alpha Q functions via secondary messengers, which are mainly lipids as well as calcium. Whereas here, they function via the adenylate cyclase. Okay, so these are the 3 subunits that we want to talk about, the alpha subunits, G-alpha S, G-alpha I, and G-alpha Q. Now, all of them, these G-proteins function via enzymes, and what they do is that they utilize secondary messengers. And secondary messengers are small molecules. They include cyclic AMP, cyclic GMP, calcium ions, IP3, inositol 1,4,5 triphosphate, and diacylglycerol. Now, the thing is, why is it that cells use secondary messengers? Why are they good? Well, they're good because they are small. They can diffuse to other compartments freely. The signal can be amplified, and this is a very important aspect in the endocrine as well as cell function overall. Now, remember that we have very little amount of hormones in our system, and the system, in order for the system to be efficient, the signal must be amplified. This little hormone must do humongous, large effect. So the signal must be amplified, and secondary messengers give a sort of like it facilitates the ability of cells to amplify signals. Also, now secondary messengers can be used in multiple pathways. So you can have a single hormone, and we'll talk about this maybe at the end of this lecture. You can have the ability of cells to integrate or to activate and inhibit and regulate different pathways all at the same time using the same secondary messenger or the same receptor as well. And this is an important aspect. Now, the thing is, and this was like an argument, not an argument, a discussion that I had with Dr. Nafid Abu Tarbush, and it was about how is it that a cell knows that it has to activate this pathway or that pathway if they use the same secondary messenger and the two pathways are regulated by the same secondary messengers. And he says something like it's all about fine-tuning. That is, it depends on the extent of activation of the receptor, the activation of the G protein, the activation of the enzyme, adenylyl cyclase, for example, that produces this secondary messenger. Now, so it's all about quote-unquote fine-tuning. Now, in my opinion as well, there is another mechanism, and that is it also depends on the use of other secondary messengers in different pathways. So you can have an enzyme that can be upregulated by a certain, not only by one metabolite, but it can be also induced or regulated by more than one secondary messenger or more than one regulatory molecule. An example is phosphofructokinase, which can be regulated by the fructose 2,6 bisphosphate, as well as citrate, as well as ATP and AMP, and so on. So this is like outside the lecture, just to give you a flavor of how sophisticated the system is. Now, remember how we classified hormones according to different aspects, like their solubility, for example. But well, we can also classify hormones according to, and the receptors according to what secondary messengers they use. Now, this is not for you to memorize, but you need to look at the main ones that we talked about in details. Okay, so we have these receptors that regulate, or these hormones that use cyclic AMP as secondary messengers. We have these other hormones that use the phosphoinositides, the IP3 and calcium ions, and so on. And we have these receptors that use receptor tyrosine kinases. So, you know what the overall signal transduction pathway is. The ligand binds to the receptor is activated, the 7 transmembrane receptor is activated. This activates the G protein. The G protein activates, for example, adenylyl cyclase. It increases cyclic AMP, the secondary messenger, and you have an effect that takes place in the cell. Okay, so I'll give you an example. We have adrenergic receptors. These respond to epinephrine, and epinephrine is the hormone responsible for fight or flight response. It belongs to the catecholamine hormones. They're known as catecholamines because they have this ring right here. Okay, so what happens is that this epinephrine binds to beta-adrenergic receptor, and notice that when it binds to the beta-2 adrenergic receptor, you have relaxation of smooth muscles, you have inhibition of insulin release, you have stimulation of gluconeogenesis, glycolysis, we have activation of lipolysis. The purpose is to produce a lot of energy. Again, fight or flight. Now, what's really interesting is that epinephrine can also bind to the alpha-1 adrenergic receptor, and it has an opposite effect. It causes vasoconstriction of blood vessels. It induces glycolysis as well as gluconeogenesis. So here we have, again, there's a video that you can watch. I hope that you'll be able to watch it. So again, the idea here is that the ligand binds to the receptor, the alpha, beta, and gamma subunit. The alpha is released. It travels through the membrane. Again, this is an efficient system right here. Instead of the receptor moving in a three-dimensional area, and it all depends on random collision of molecules, rather you have the subunit, the alpha subunit, moving in the membrane. So it makes it easier for the alpha subunit to find its target, which is adenylate cyclase. Once you have this interaction, you have production of cyclic AMP, and cyclic AMP can bind to the effector molecules. Now, what's important about the alpha subunit is that it is not an enzyme, but it does have an enzymatic activity, and it's an intrinsic enzymatic activity. It acts on itself. It acts on the molecule itself. This enzymatic activity is GTPase, and what it does is that it hydrolyzes GTP to GDP right here, and that allows the alpha subunit to rebind to the beta and gamma subunits, again inhibiting that. Again, this is termination of the signal, and that's important so that you can have another effector that can bind to the molecule, another effector that binds to the molecule, activating it. So you need to terminate the signal and reactivate it again. Now, remember, this is the alpha subunit. What it does is that it inhibits the molecule. All right. Now, what are the cellular effects of cyclic AMP? Well, these are the ones: degradation of storage fuels. You have secretion of acid by gastric mucosa. All of these things. Now, let me just focus a bit on this. This is a nice piece of information. So, why does it increase the secretion of acids by gastric mucosa? Because why does coffee increase acid in gastric mucosa? Why is it that it can induce ulcers or it can cause ulcers, for example? And some people have their stomach annoying them with acids. The reason is that this is a nice study from 2017. Caffeine actually induces gastric acid secretion via the activation of adenylate cyclase and activation of adenylate cyclase and increasing the level of cyclic AMP, and that's what induces the secretion of acid by gastric mucosa. All right, then what happens? Again, there's another video that you can watch, I hope. What happens is that cyclic AMP binds to one molecule in particular known as protein kinase A. Now remember, it depends on the cell type, okay? So it activates protein kinase A. You have dissociation of the catalytic subunits of the protein kinase A, and that induces protein kinase A can phosphorylate many proteins, including glycogen synthase, inhibiting it, and activating the phosphatase the phosphorylase, the glycogen phosphorylase, eventually inducing breakdown of glycogen and glycolysis to generate energy. Okay. Now, signal amplification, again, is an important aspect of the signal. So you have very little hormone present in the body, but its effect is humongous because of that, signal amplification. And the idea is that one molecule of epinephrine can activate, let's say, one receptor, right? Now, the same molecule, epinephrine, can dissociate, and it can bind to another receptor as well. So let's say that we have activation of one receptor. This one receptor can activate multiple G proteins. And these multiple G proteins can activate, each one of them would activate a single adenylate cyclase. Now, adenylate cyclase can produce thousands of each one of them can produce thousands of cyclic AMP molecules. These cyclic AMP molecules, each one of them would bind to the protein kinase A, so there's no amplification here, dissociating the catalytic subunit, and protein kinase A can phosphorylate many proteins, and these proteins by themselves can be, these proteins themselves can be kinases as well, or enzymes that can produce, again, thousands of other molecules or target molecules, or they can target many molecules. And finally, you have the production of thousands of products from each one of these enzymes. So you have multiple levels of amplification of that signal. Okay. Now, the amplification as well can occur at the level of hormones themselves. Remember how we have hormones secreted from the hypothalamus. They can affect the anterior pituitary gland, and these hormones can be released as well from the anterior pituitary gland, and they can affect other target glands. Well, you can have one hormone resulting in secretion of hundreds or thousands of hormones from the anterior pituitary gland. Each one of them can target a certain gland or certain tissue, again producing hundreds or thousands of hormone molecules again. So we have multiplication that also takes place at the hormonal level. All right. So how about termination? How is the signal terminated? Remember, if it is not terminated, you would have something known as desensitization. So how is the signal terminated? Well, one mechanism is dissociation of the hormone itself. Once it's dissociated, the receptor becomes inactivated. The second level is the GTPase activity of the alpha subunit of G protein. We talked about that before. So converting GTP to GDP, hydrolyzing GTP to GDP. We also have hydrolysis of cyclic AMP, the secondary messenger, by another enzyme known as phosphodiesterase. So we have adenylate cyclase converting ATP to cyclic AMP, and we have phosphodiesterase converting cyclic AMP to an inactive molecule known as AMP. And then we have also phosphorylation of the receptor itself by the receptor kinases. And when the receptor is phosphorylated, it's bound to a protein known as beta-arrestin. And beta-arrestin makes the receptor, even if it is ligand-bound, it makes it inactive. So you can see the light. When there's light, it means that the receptor is active. Okay? So arrestin inactivates the molecule itself, the receptor itself. So there are, again, different levels of regulation as well. Here's a nice example that you should know about. What's the effect of cholera? Why is it that cholera toxin? Well, the thing about cholera is that it induces continuous diarrhea, and that causes dehydration, and it can be fatal. It kills people. So how does the cholera toxin function? Well, what it does is that it keeps the alpha subunit of the G protein in the active state. Right? So it inhibits the GTPase activity of the alpha subunit, and that makes the alpha subunit always active, constitutively active, producing a lot of cyclic AMP. Now, the idea is that when cyclic AMP, when adenylate cyclase is active because of the alpha subunit, and you have production of a lot of cyclic AMP, that causes the flow of sodium ions as well as chloride ions out from the mucosa. And this happens continuously, and that results in diarrhea as a result. Okay. There's another pathway. So we have a G protein pathway that relies on adenylate cyclase and cyclic AMP, but we have another pathway that utilizes G alpha Q that activates, that results in the production of different secondary messengers, namely IP3 and diacylglycerol. So it's the same idea. You have certain hormones that bind to 7 transmembrane receptors, activation of G alpha Q, and that causes activation of an enzyme known as phospholipase C. What phospholipase C does is that it cleaves this molecule right here, which is phosphatidyl 4,5-bisphosphate, also known as PIP2. So you have 2 phosphates right here in this molecule, 1 and 2. Okay? So what happens is that phospholipase C cleaves this molecule, producing 2 molecules. One of them is known as IP3, and the other one is the diacylglycerol, which is present right here, diacylglycerol. Okay? So di, because there are 2 fatty acids attached to the acyl group right here in this fatty acid, in this molecule on glycerol. Okay, so we have these 2 molecules, and they both act as secondary messengers, and again, they work in tandem. That is, they work together. They don't work by themselves. So what do they do exactly? Here's phospholipase C. It has different domains. We defined domains before. One of them is a G protein interaction domain. It's responsible for interacting with the alpha subunit. So when the G protein is activated, it can bind to phospholipase C. Now, notice something again. We have this molecule attached to the plasma membrane, again, to facilitate or to speed up the interaction, to facilitate the interaction of different molecules and to speed up the signaling pathway. All right. So phospholipase C binds the alpha subunit, gets activated, and it can convert PIP2, which exists in the plasma membrane. It's part of the plasma membrane. So what does IP3 does exactly? Well, let's look at here. So you have ligand binding to the receptor. The receptor is activated. It activates the G protein. The alpha subunit is released and it's active. It interacts with the phospholipase C. And when it does, it converts PIP2 into IP3 right here and diacylglycerol. Now, IP3 is released from the plasma membrane and it can interact with a receptor, with an IP3 receptor that exists on the endoplasmic reticulum. This receptor forms a channel allowing for the release of calcium ions from the ER to the cytosol. This is fascinating because what happens is that you have the production of diacylglycerol from phospholipase C. Now, diacylglycerol, what it does is that it interacts with other proteins, other molecules, regulatory molecules, namely and specifically protein kinase C, activating it. But activation of protein kinase C does not only depend on interaction with diacylglycerol, it also depends on interaction with calcium ions, which come from the ER. So I said they work in tandem, they work together, this IP3 and the diacylglycerol, because IP3 releases calcium, and together with the diacylglycerol, they activate protein kinase C. Now, protein kinase C can then activate phosphorylate many proteins, activating them or inactivating them, resulting in a certain effect. To cover exactly what happens with protein kinase A, which is activated by both diacylglycerol and calcium ions. Well, if you look at the structure of protein kinase C, it has multiple domains in different shapes and colors, as you can see here. And you have these domains right here, the C1A domains. Now, one of them, C1A and C1B. Now, one of them requires diacylglycerol, or it uses diacylglycerol to interact with the membrane. And you have another domain that requires calcium. Once calcium is bound to that domain, well, and you have the other one associated with diacylglycerol, protein kinase A associated with the plasma membrane. Protein kinase C, if it is inactivated, you have, in the end terminus of that molecule, you have a sequence known as pseudo-substrate. So it looks like a substrate, but it's not a substrate. It's a pseudo-substrate. It's a fake substrate. What this does is that it covers the active site of the enzyme, so it's not active. But when it associates with the plasma membrane via diacylglycerol and with the help of calcium ions, what happens is that the diacylglycerol pulls out the pseudosubstrate from the active site. Now, the active site is free, and protein kinase C can phosphorylate many targets. In the cell, it can bind to calmodulin, and calmodulin, it modulates the functions of many enzymes. Notice what these enzymes are. You have adenylate cyclase, you have phosphorylase kinase, you have pyruvic carboxylase, all of these metabolic enzymes that are regulated by calmodulin. So we have these processes that are affected or regulated by calmodulin and by the increase in calcium. Again, you have glycogenolysis, you have the histamine secretion, insulin secretion, and so on. Again, this is not for you to memorize. Just look at them. Look at the different pathways that are regulated on different cells by the same exact hormone. So it's all cell specific. So how is the signal terminated? Again, this is really important. IP3 is a short-lived messenger. It gets degraded into inositol by two different mechanisms, but we will not talk about this. So IP3 is converted to inositol. That's one. And diacylglycerol can be phosphorylated to phosphatidate, or it can be hydrolyzed to its original components. So it can be phosphorylated to phosphatidate. That's one. Or it can be degraded into its individual components, that is the fatty acids and the glycerol. And that's how the signal is terminated. Now, notice that all of the termination signals are enzymatic, so that it's really quick. What happens to so termination is quick. Okay. So these are G proteins. We covered G alpha subunits, and we covered GQ subunits. So let's talk about the cell surface receptors now. You can take a break. So let's talk about the cell surface receptors and how they induce a signal. Now, enzyme-linked receptors are, it's a large class of proteins, and they regulate many facets of cell biology. They can regulate growth, proliferation, differentiation, survival, death, all of these things. They are regulated by growth factors that bind to them, and these growth factors exist in small concentrations in the body. Now, these receptors overall, they can have two effects. They can have a slow effect, or they can have a fast effect. Now, the slow effect is basically, it includes, it's a genomic effect. That is, it regulates gene expression. So again, it takes time to transcribe a gene and translate a gene. So it takes hours. And we have a fast response as well. This is enzymatic. So what you have is change of the actin cytoskeleton, for example, which takes place within like a few seconds or minutes. So this is a fast response. There are different types and different shapes of these receptors. Some of them are actually heterodimers, like the insulin receptor. Others are monomers, just a single chain. You have receptors having repetitive domains, and some of them have the same common domain and different numbers. But what they have all in common is that they have a kinase domain right here intracellularly. So when a ligand binds to a receptor, this kinase domain is activated, and it can activate or regulate many signals. And that's why we call it enzyme-linked or receptor-linked kinases. All right, so receptor tyrosine kinases, they phosphorylate kinases. This is, they contain, again, the intracellular kinase domain. And one aspect that is important, so this is the flow of what happens. The ligand binds to the receptor. The receptor dimerizes. Notice that how the two chains are located a bit far from each other. Well, once the ligand binds, it induces the dimerization of the receptor. So, and you have the formation of specifically homodimers. Now, the kinase domains get closer to each other in this case, and they phosphorylate each other. So this is called autophosphorylation. When you have the domain phosphorylated, the presence of the phosphate groups facilitates the interaction with other molecules with the receptor. So the presence of this phosphate group is important for interaction with other molecules. Okay? So this is the pathway overall. Ligand binds, dimerization, autophosphorylation, association with different regulatory molecules. So here's an example, insulin and the insulin growth factor receptor. The insulin receptor is a heterotetramer, it's a heterotetramer. Again, same idea, ligand binds, dimerization, and autophosphorylation, then association with different proteins. So autophosphorylation activates signaling by first phosphorylation of the tyrosine that makes the kinase activity of the receptor more active. That's one. And two, when the tyrosine residues are phosphorylated, it allows for interaction with other molecules, including the insulin receptor substrate and GRB2. So these are docking molecules. Okay. Two responses. We have an immediate effect, and we have a longer lasting effect. The longer lasting effect, or the slow response, is basically genomic. You have increased expression of certain genes like that synthesize glycogen or triacylglycerols. And we have the immediate or fast response, which takes a few minutes. Here you have increase in the rate of uptake of glucose from the blood, modulation of certain enzymes, and these effects do not require protein synthesis. All right. So we have two pathways. What insulin receptor does when activated is that it activates two pathways. One of them is dependent on RAS, and the other is independent of RAS. Now, what RAS is, it's a G protein. It's a small monomeric G protein. So it's different than the large trimeric G protein, the alpha, beta, and gamma. It's a small molecule. It's regulated by binding to GTP and GDP. So if it is GTP bound, then it is activated. If it is GDP bound, then it is inactivated. Now, both of these pathways, whether RAS dependent or independent, depend on the IRS1, the insulin receptor substrate 1. So that's the RAS dependent pathway. What happens is insulin binds to the receptor, again, dimerization, autophosphorylation, and that allows for interaction with a molecule like GRB2 right here. Now, GRB2 binds to the receptor. Actually, it binds to the IRS1, and then it binds to GRB. Okay, so it's not shown in this figure. So IRS1 and then GRB2. Now, GRB2 binds to a protein known as SOS. What SOS is that it is a GTP exchange factor, meaning that it promotes the release of GDP, allowing for the triphosphate molecule to bind to functions on RAS. When RAS is GTP bound, it can activate a kinase known as RAF, which activates by phosphorylation another molecule known as MEK, which phosphorylates and activates another molecule known as ERK. ERK is a transcription factor or a co-regulatory molecule. It goes into the nucleus. It binds to other molecules, other transcription factors, inducing transcription. So how about the RAS-independent pathway? Well, it also depends on the presence of IRS1. IRS1 binds to the receptor when it's activated, when it's ligand bound. So you have IRS1 right here, and then you have interaction, then you have activation of PI3 kinase, resulting in the production of phosphoinositides. Okay? So once you have phosphoinositides formed, that leads to the recruitment of a kinase known as protein kinase B to the plasma membrane. Now you have these phosphoinositides on the plasma membrane. So you have recruitment of protein kinase B to the plasma membrane. They interact with the inositides on the cell surface, and it changes the conformation of the receptor. And once it's activated, then it can phosphorylate many proteins like glycogen synthase, kinase, activating glucose uptake as well as glycogen synthesis. Okay? Now, one target for this receptor is activation of phospholipase C, and you remember what phospholipase C does. Conversion of PIP2 into IP3 and diacylglycerol. So you have the activation of multiple pathways by the same ligand and same receptor. Okay, so this is the crosstalk that I talked about. You can have a single molecule or bind a single ligand binding to its receptor, and this receptor can activate multiple pathways like phospholipase C. GRB2 is recruited to the receptor, activating the RAS pathway. And then you have the activation of the PI3 kinase and eventually the PKB. Okay? And you have different effects. This is all balanced. This is all coordinated within the cell, so that you don't have activation of one pathway only. Finally, how are the signals terminated? By phosphatases. Okay, so what these do is that they remove the phosphate groups on the receptor, or the phosphate groups on the other molecules that are phosphorylated within this pathway by any one of these kinases. I'm not gonna. It's really hard to talk about all of the phosphatases and all of the kinases and all of the regulatory molecules, but this is the take-home message. Okay, so here's a joke. You can read it, enjoy. Allah معكم. Hope you enjoyed it and you don't have a headache. ورمضان كريم. كل عام وانتم بخير. السلام عليكم.